Hi, everybody. Welcome to our 21st and possibly last roundtable as a webcast since the start of this pandemic. I'll have more on that at the conclusion of our program today, but about our first live roundtable since the pandemic began and what we're going to do to make it accessible to absolutely everybody. But before that, we have a real treat in store for you today. At the biannual International Myeloma Workshop held recently in Vienna, Austria, this is a time for top experts to come together and share the most up-to-date myeloma treatment and information that clinics and physicians use today. So doctors reported on their experiences with monoclonal antibodies and antibody and peptide drug conjugates. They had information there about updated recommendations for induction therapy or consolidation and maintenance therapies. And soon we'll have the annual American Society of Hematology, or ASH, meeting in Atlanta, where we'll get results and updates on ongoing clinical trials and a sense of where the future myeloma treatment might be heading. So we thought now would be a great time to discuss the big picture of myeloma research and treatment and what it might mean for each patient. And for these insights, we couldn't be more fortunate to have Dr. Fonseca and Dr. Ola Langrens with us today. Now, I met both of them about 9 to 10 years ago, and they've been key to the growth of the HealthTree Foundation and the myeloma crowd since the beginning. They both serve as scientific advisors for us, and they help set the direction of our programs. Most importantly, I think they've become very trusted friends. Now, Dr. Fonseca has long been a valued member of the Mayo Clinic staff in Scottsdale, Arizona. He's currently the interim director of the Mayo Clinic Cancer Center over all Mayo Clinic facilities, as director for innovation and transformation relationships at Mayo Clinic in Scottsdale. And I'll never be able to thank him enough for his guidance to helping create the HealthTree Cure Hub, which we'll mention today in our discussion. And this is a very exciting time also for Dr. Ola Langrens. So since last November, he's been putting together a world-class team of researchers and clinicians as director of the myeloma program at the University of Miami's Sylvester Cancer Center. Prior to that, he was at Memorial Sloan Kettering in New York, the NIH in Bethesda, Maryland, and the Karolinska Institute in Stockholm, Sweden. So both Drs. Fonseca and Langrens are considered to be among the most forward-looking and innovative minds in myeloma, and this promises to be an exceptional program today. Now, to learn more about our speakers, you can see short biographies of the faculty together with an agenda by our roundtable program director, Greg Brozite, for whom I'm very grateful for putting together these amazing programs, both live and virtual. So thank you, Greg. And should you have any questions to submit as you think of them, you can click the Q&A link at the bottom of your screen. And it's pretty tough for the speakers to address really personal types of questions. So try to think of questions that would be most appropriate for everybody. So now I want to thank our generous sponsors: Amgen Oncology, Adaptive Biotechnologies, Bristol Myers Squibb, Genentech, GSK, Janssen Oncology, Karyopharm Therapeutics, Oncopeptides, and Takeda Oncology, because without them, we cannot provide this valuable and informative program for patients throughout the world. Now, part of the agenda. For the next 45 minutes, we'll discuss the latest general ideas in myeloma research and treatment, kind of where we've been, where we are, and where we hope to go. And we'll follow that with 45 minutes of Q&A. So go ahead and enter those, like I said before, at the bottom of your screen. So I think we should just get started. And thank you, doctors, so much for giving us your time today.
Good morning, Jenny. Thank you for having us.
Yeah, so fun to see you both. And it's it's been too long since everybody has seen each other in person, doctors and patients alike. So
hi, Jenny. I say a good midday here. It's 12:30 in Miami. I think it's morning where Rafael is in Arizona. Is that right? Good morning, Rafael.
Good morning.
Good day to you both. Okay, let's get started with a few questions. And because it's going to be a broad discussion, I just want you to both jump in with your opinions about these different topics. So when we first organized that first scientific advisory board meeting, our focus was really meeting unmet needs in myeloma and see what we could do to address some of those. That's why, for example, we created that Myeloma Crowd Research Initiative about high-risk disease. So looking back over the last maybe decade or so, how has the research changed since we first met and now what is still considered an unmet need?
Maybe I can take a first stab, Jenny. I think, the research continues to move forward, but unlike what we have seen in the past, where there were two tracks. One was understanding the disease and the biology, and then the other track was understanding the treatments and, what treatments may work better. I think what we have seen is a couple of things. First is the pace of the research has greatly accelerated. And I'm sure we'll talk about this, but particularly with the treatments along the lines of immunotherapy, that has been a major change forward. But the second thing is that those two streams have come together quite nicely, so that as we think about the biomarkers, we think about the classification of myeloma, we think about how the treatments work in the different subtypes. I think everything is merging in one common thread. So I think it's good, it's exciting. It can be daunting because there's a lot of information, but it's all moving in the right direction.
Mm-hmm. Great. And Dr. Landren, what are your comments?
I think Rafael hit all the high points. I think to me, the main big thing is really the pace of how fast things are changing in the good direction. And I think it's going to go just faster and faster. I think looking at the pipeline of all the new drugs that are coming, there are 25, 30 or more drugs in development. I think we will probably have a few drugs approved every year for several years to come. Many of these drugs are better than anything we have ever seen, and better than all the FDA-approved drugs, if you just look at the overall response rates from available data. I also think that the testing for response evaluation with MRD testing is going to help speed up drug approval, because if the FDA finally will endorse that, that means that drugs can be approved based on MRD status, which will basically give access to drugs years faster than we currently do. And it will also probably help in clinic to steer the intensity of therapy, to turn on and turn off therapy, or scale up or scale down therapy. So there's a lot happening right now, and it's going faster and faster. I agree with Rafael. Therapy and biology is getting closer, and that's a good thing.
Mm-hmm. Absolutely. Can I ask a follow-up question to that? Because I think maybe 10 years ago, you may have wanted the FDA to make approvals in a different way. And what I've seen in myeloma, and maybe this is just a patient perspective and not your perspective, but I've seen them be very aggressive in the myeloma space about trying to get things approved as fast as possible. Do you have any comments about and that usually you have to go through the phase 1, the phase 2, and the full phase 3 before something gets approved, and you're not really seeing that anymore. You're seeing some approvals after phase 2. So do you want to discuss that topic?
Sure. Go ahead.
Go ahead, Lola.
Mm-hmm.
No, you go ahead first.
I was going to say, I think you're going to find a variety of perspectives on this, some of which may even be philosophical. I personally like the idea of fast approvals for a number of reasons. I mean, first, the faster you can get the drug in a way that we can prescribe it, of course, obviously, the faster we can use it for patients that we think will benefit from that. I think the idea of how we used to do research and require certain things to show that a drug was worthwhile have become incredibly hard for myeloma. And that's good, because things such as overall survival, which has been historically a very important way to measure the value of a drug, it's very hard to measure now, because fortunately we see a longer and longer duration of disease control, which relates to better survival. And at the end of the day, doctors and patients find ways by which they can actually better use the drugs once they're released. And there's a lot of learning. And we know that with subcutaneous Velcade, we know that with, all the drugs that have been used for the treatment of myeloma.
Mm-hmm.
I think in response to your question, Jenny, I do think that still the different phases still apply. The phase 1 designed to identify what's called the toxicity profile of the, the camera turned off, both Rafael and me. So the phase 1 studies are here to identify the safety and also identify the right dose and the dosing schedule. That does not change. That always has to be done when a new drug is being developed. The phase 2 studies are the next phase, when you are usually doing a single arm study. All patients are treated the same way. It feeds off the phase 1 study. And this is now designed to see if there is really a true signal in terms of efficacy. You continue to capture information on the safety side. And traditionally, the phase 3 trials, they are the randomized large studies that then would compare the therapy to a identified control arm. And if you show superiority, you live up to your endpoint you have stipulated, then that would be equal to granted a full approval by the FDA. I think the new era you're alluding to, Jenny, is really, I think, what Dr. Richard Pastor at the FDA has created. He is an academic physician. He came from the MD Anderson to the FDA with a lot of clinical experience and as an academic doctor. And he really has transformed the FDA in a very, very, very sophisticated way. So they are very fast and they're also really, really good. And they have looked to see how could they sort out if there are promising drugs earlier and give them an accelerated approval. But still, an accelerated approval has to be followed by the full work with the randomized trial to give the full approval. So accelerated approval gives faster access, and then the full approval leads to the full approval. But there are examples of drugs, not for myeloma, but for other diseases where there was an accelerated approval granted, and then the final study didn't deliver, and then it was actually retracted. So that's rare, but there are examples of that. So they are doing a good job, but they're basically letting patients getting access faster. It takes so many years to do these large studies.
Right, it does. So yes, it's good that there's a stringent process, but it's also good that things are happening at an accelerated pace. So for both of you, and what makes you more optimistic and hopeful? So when a newly diagnosed patient enters your office today, and what tools do you have available that you didn't 5 or 10 years ago, or even 15 years ago?
You want to start, Ola?
Yeah.
Sure. Well, maybe we take turns here and starting, and we can go back and forth. And so, I mean, first of all, look at all the drugs we have access to now. 10 years ago or more, when I started off, we had a VAD regimen between Cristin, Erythromycin, and dexamethasone. These are drugs that we no longer use. So we used to give, say, 3 cycles. If you gave more than 4 cycles, that was really cardiotoxic. So giving 3 cycles of that was kind of what we did. Almost none of the patients achieved complete response. And melphalan chemotherapy with autologous stem cells was the best that could be done to enhance the effect. But now we have all these new drugs. We have 4 drug combinations, antibodies with the PI-IMiD steroids. And there is also work going on right now looking at maybe that could be even antibody-only therapies or CAR T cells going up front or things like that. So there is a lot of change, but we already have the 4 drug combinations, and we also have these MOD tools to evaluate responses. So these are really the big changes we already have, but all the other drugs that are coming and the changes, I think, are going to transform the field even more.
Great. And Dr. Fonseca?
Yeah,
diagnosed patients?
I think there's a few things that are within the horizon, I think. And what I'm going to say, I have thought, a lot about saying, but I think we can be factual in that I think we're in the brink of having proportion of patients that it's measurable and hopefully growing that will be cured from their multiple myeloma. So again, I don't say this lightly, but we will only know that unfortunately many years from now. It's like, it's like looking at the stars far away, that you're looking at the past, so that we don't know right now what this will mean as we go into 5 and 10 years from now. But when you look at patients who are treated with the best that is out there, so like some of this 4 drug combinations, and we see that patients can achieve an MRD status in 60% of patients, and some of them seem, the ones that particularly seem to be sustained, it is hard not to believe that some of those will be permanent solutions for the person. So my hope for those that are currently what we consider the transplant eligible population, we'll start seeing some of that convert into just permanent control of the disease. In the transplant ineligible patients, I think we're seeing big strides, and probably the most important one, and we can talk more about this later, is the MAIA trial with a combination of the daratumumab, lenalidomide, and dexamethasone. And for that population, the results have surpassed anything we could have ever done in the past, including if you were to treat those patients with stem cell transplant. So I think for someone who's diagnosed with myeloma, who's now today over the age of 75, and you look at the actuarial tables of survival for us in the population, and if I were to ask myself when I'm 75, on the average, how many years I have ahead, we're not quite there, but we're getting close to say that the survival is going to be close to what would be the normal life expectancy as well too. So again, the best would be we never diagnose patients with myeloma, perhaps even better. Or if we can't do that, we prevent myeloma. But in the meantime, I think those are the big strides that I see coming forward.
Mm-hmm. And, contrary into that, I guess, is just that for certain patients, that those great outcomes aren't the case because we, like what you were saying, Dr. Fonseca, maybe you want to start with this. You recently conducted a study about patients who reach certain lines of therapy, and as many as 8% of newly diagnosed patients never even make it to their first line of therapy. So what are some of the difficult questions about patients like these or in the high risk area that are influencing what your research is doing?
Sure. Well, and what I say is pretty conceptual, and that's at large, right? But we all live in the real world. So there's people that for a number of reasons, be that because of the type of myeloma they are diagnosed with and what we call the biology, so that is the behavior of those cells and how those cells, react or not to chemotherapy. But also other factors, it's just like what we are seeing with COVID now. If you go into myeloma and you have heart disease and you have lung disease, or you already have, significant handicap because the lack of good kidney function, those things are against the person because, it's a bit of a combination of things. It's harder to move, it's harder to eat, it's harder to tolerate the treatments and so forth. So there will always be a whole range of how people react to and are able to respond to the treatments and also to stay on treatments. I always like to repeat this often. We are in the real world. So we can read a study and say, well, in this study, they did this and that. But when we're in the clinic, we know everyone is different and we have to adjust, sometimes pause, and we have to do the best with what we have to try to make sure that in a safe way, the person can get as much of the treatment that we think is reasonable.
Mm-hmm. And Dr. Landren?
Well, again, I think Rafael covers all the bases here. I agree on everything he said. I think also maybe adding additional perspectives where I see these things kind of are moving. There's obviously the race between figuring out more of the biology and coming up with better combinations and develop new drugs. So we are already at the point where we have really, really good drugs for the vast majority of patients. I think there are some patients, but we still don't know how to treat them. And the best we have to offer is basically the same therapy as we give to all the other patients. So that's a huge unmet need, but we need to study more and better understand why the standard therapies don't work there. In the other group, I think there could probably be patients that could be treated with less therapy. I see that as a huge important task going forward and try to individualize this therapy more. So I kind of add up on top of what Rafael just brought up.
Mm-hmm. That's a really important concept. I think we'll talk about that a little bit more later too. Now, I'm hearing a lot more. When I first was diagnosed, I heard very little about real world evidence or real world data. And it was kind of like poo-pooed a little bit. It seemed like it wasn't quite as good as the clinical data that was coming out of certain trials. So, but I've heard a lot more lately about the value of it. And so could you both please address, and maybe Dr. Landren, you want to start, what real world evidence is, and then what's the potential also of using our tool, HealthTree Cure Hub, to be in gathering real world data, and how do we use real world evidence to propel research forward?
Yeah, so I think that there is more and more interest, as you allude, Jenny, to real world data, meaning patients that have been treated outside clinical trials. We all know that the clinical trials have a lot of strengths. The randomized trials have the strength that every other patient on average would be treated on one of the two arms in a two-arm study. So there shouldn't be any bias that you put one patient on the better therapy and another patient on the less good therapy, or vice versa. It's done by random. And that's why those studies are considered to be gold standard for drug development. But then you have all the inclusion criteria, people who have renal failure, people who have certain comorbidities. There could be many reasons why an individual could not go on a trial, lab markers and so forth. So for that reason, they are not representative of the average patient. In fact, there are a lot of factors that really don't add up. Like there could be age discriminations. They could have enforced all these rules. And you could argue back and forth, shouldn't they include more broadly? But then the drug development side would say, if we do that, that could also potentially make the results less strong. And then the drug may not be FDA approved. And then no one gets access to this therapy. So that's a balance there. So I think what's happening right now is that people are thinking, could we use some of the real world data to balance that towards data in single arm studies? So there is not yet any precedence to use this as the gold standard, but there have been some drug approvals for myeloma where the FDA had paid a lot of attention to real world data. One example, for example, was selinexor. So selinexor was approved similar to most of the other drugs on a single arm phase 2 study. They looked at overall response in patients who were heavily pretreated. They then compared to, for example, the Flatiron database, which is a New York-based initiative. They have information on many, many patients. And the way they did it is that for every patient treated on the phase 2 study with the drug selinexor, they looked in the database for people, for say patient number 1 on the trial. If it was a man, they looked for males, and then they looked for the same age plus minus a few years, and they looked for all these other variables that are important for biology in the big database. So they could now compare these individual patients with all these individuals that matched up. And then for patient number 2 in the trial, they did the same, and patient number 3 and 4 and 5 and so forth. So for every person, if there were some controls in the big study, the Flatiron database, you could now have basically a digital control group. So you could think about this more and say, couldn't this really be a way to do all the trials? Why couldn't it? So if you had information on patients with the disease, do you have to do randomized studies in the future? And right now, the FDA would say, yes, you have, because that is currently the standard. But being a little bit bold, maybe in the future, things could change. And the reason I say that is because this week, I think on Wednesday this week, I'm sorry, my camera keeps on turning off here, but on Wednesday this past week, the first artificial intelligence diagnostic tool was FDA approved. And this was based on the company called Page AI, also New York based. They actually got approved by the FDA a digital tool to set diagnosis of prostate cancer. So the computer could go through thousands and thousands and thousands of pictures and then learn how to set the diagnosis. So if you now put a new single tissue in front of the computer could say, this is prostate cancer or not. Oh, so wow. The computers are now using digital controls to say cancer, no cancer. So why could we not develop drugs like that? I think this will probably happen at some point. I don't know when, but I think it probably will.
Mm-hmm. Fascinating. Okay, yeah, I haven't thought about it like that before. What a great idea. And Dr. Fonseca, your comments?
No, I think Dr. Landren has covered the highlights to as well there. In reality, in medicine, we're far behind. Every other industry is already using real world data, right? I mean, you tell your friend that you may be thinking about having a baby, and the next thing you know is you having ads for diapers in your feed. I mean, that's how the real world works for everything else. You have the gathering of all this information, and we proceed with that. I always think that there is a future where we will be able to use this, especially for control arms, because people say, well, with randomized trials, what you move away is from the bias that you say, I'm going to treat this person because maybe I think they're a little bit healthier, even if it seems like their kidneys are the same and the liver is the same, etc. There still is a possibility you see the person and you say they look stronger to me, and for that reason the group that is going to get, say, a transplant is going to be stronger. But the larger the data sets, the less likely you are that those bias will come into play, and then will bias that. So you could say, if you develop a clinical trial where your eligibility criteria is super simple, like age over 60, and then you have creatinine this and a diagnosis of myeloma, and then you treat with a drug, then you could compare that to a control arm that wouldn't be 200 patients, but could be 10,000 patients that are the standard of care treatment in the community. And then you would know for sure, especially if your eligibility criteria are so simple. This reminds me of an Argentinian writer, Borges. He wrote a little story that was called The Cartographer, and the cartographer was the king, told the cartographer to draw a map of the kingdom. And the first draft was not enough, so he was sent back to the drawing board and back to the drawing board and back to the drawing board. And in time, the map became so good that the king and the cartographer could not tell what was reality and what was map. And I think that's what's going to happen as we move with real world data. We're going to see that in the real world, what happens with the medicines, and it's going to be as if we are doing the clinical trials, as we collect this data. So I'm a big fan of real world data. I think you're right that initially it was poo-pooed and people said, no, the only way to do it, if you made the orthodox way of doing it, would be through randomized clinical trials. But we're learning a lot about cancer because of real world data sets.
Mm-hmm. Yeah, it's fascinating. So in the clinical trial environment, but also outside of that, because we've been running some just data sets, on like genomic studies and things like that, looking at 414 patients first and 1114 patients, kind of subdivide it. And we're able to get like a bulk of enough patients. So you have enough data so you can start seeing patterns. So it's just, and maybe a study that might not be funded otherwise.
Well, and let me compliment Jenny. I forgot to say, so the HealthTree is phenomenal for this because, let's say you run a clinical trial like we were talking about selinexor for approval. But let's say that for whatever reason, loss of taste happened in 1% of the population. You're very unlikely to report that in your FDA, reports. But as you build these data sets and like people, that enrolls in, say, HealthTree, you could even query, has anyone else lost taste? And then you might find out, you could even ask everyone. And it turns out only the ones, and I'm making this up in case, just total made up thing, but you could say, oh, maybe the ones that are getting whatever drug are the ones that are losing their taste. So that's one way by which you're going to be able not only to generate the data, but to go back and query it in a way that you can pull out things that are not apparent right from the beginning. And yeah, as we were saying before, you learn a lot as we go forward with these new drugs on how to best use them, what to expect, new side effects, etc.
Mm-hmm. Okay, so interesting. Now, Dr. Landren, you mentioned before about MRD testing and trying to shorten up this clinical trial development process, because now that patients are living so long, if you really went through the whole process, you'd be waiting 8, 10, maybe more years to see if your clinical trial would read out. So I think what you're saying is you're trying to use MRD testing to kind of shorten that up. And some I know I've heard that it's integral to determining if patients are potentially being cured or having better or projecting out better outcomes. As two of the world's leading experts in MRD testing, what do you both see as the most immediate opportunities for the future for MRD and other testing like that? And Dr. Landren, you can start.
Well, I do think there will be many different deliverables. I think to simplify, I think there are two main tracks that I see in the near future. The first one is the regulatory track. And that is really if the FDA and other regulatory agencies around the world would acknowledge MRD as an endpoint that could be used for drug development. We already touched on this. If you have a randomized study, you show arm A versus arm B, arm A being the experimental and arm B the control arm, that there is a higher rate of MRD negativity. And if you capture that after 1 year, you have now the data to see if there is a superiority in MRD negativity. And if there is, that could grant approval. In the current setting, the standard would be to capture progression-free survival. And with drugs working very well, which is fantastic for patients, for drug development, that means that it's going to take a long time to wait for the data to be mature. So typically you look for a median progression-free survival or at a fixed time point. So 5 years could turn into 1 year to have the data. So if that's true for every drug, if we think about how these drugs are developed, the whole field is a cascade of consequences. So this would be to put on all the jets and just speed up drug development. That's huge. The other of the two is really, I think, is the opposite direction is the clinical utility. And I think physicians right now hear about MRD. We know that 80% of patients with myeloma are treated in a private practice that sees a lot of different disorders, and they don't have access to all these different tests. Not all the big centers in America do have, and worldwide it's the same phenomenon. But there are centers, Rafael and I have worked on it for a long time. We have access to it, and we would look at the results. Some of us use it for decision making, although we can argue that there is not formal data to really say how it should or shouldn't be done. But it's not different from a lot of other things we do for M spikes or PET CTs or things. So that's like a long discussion. But I think if there were blood-based MRD tests that are reliable, validated, available everywhere, I think that would be the big game changer also. I think that people would use that to maybe hold back on transplant, maybe if it turned from negative to positive, maybe start some relapse therapy earlier. If patients sustain negative in the blood longer, maybe having breaks from the therapy, treatments, holidays, and maybe patient could eventually go off therapy if therapies are great, which I think they will. And then, of course, you could monitor with blood tests. So I think the convenience and the sensitivity of MRD would really transform the field in the clinic. So these are two different avenues. And I know that this is a little bit futuristic, but I think this is going to happen sooner than we think. I don't know what Rafael thinks about this.
Yeah, I'm curious, Dr. Fonseca, about when you think this could happen, because that would be great.
Okay, yeah. So let me use the same tracks that all I use. As far as the FDA, I would say is long overdue. There has never been a determination of anything in myeloma that has the power to allow us to predict what will happen into the future as MRD status is. I devoted all my life working, as you know, to FISH and genetics, and you know, we were all very excited and everyone was happy, and that was quickly embraced as an important, you know, biomarker. But the power that MRD has to be able to predict long-term outcomes, and this is not 100%, nothing is 100%. But when you look at the ability to discern outcomes, really MRD can accelerate our view into that future. So if that were to be translated into shorter drug development, faster access to drugs, also, one would argue if you have more drugs and they're developed faster, then you're going to have more competitive economic, factors and pricing for drugs, because then it's not as onerous to have this drug development process. Then I think everyone wins with that. So I would say, to me, it's pretty self-evident. I mean, there's just a large amount of information that would suggest that we should be using this for approval processes. In the clinic, I think we are ready to use it for the clinic. I'll start by saying what Dr. Landren was saying. I don't know why MRD has to be treated with this exceptional approach. We've never done that for anything, for the free light chain, for the M spike. And those things are integral to how we understand myeloma. They just make our practice more informed. So there's really no reason to treat it in any exceptional way. People now have said, oh, well, sometimes we need phase 3 trials. That is what's needed for drugs, not for this diagnostic, approaches. You might say you might need it for a whole strategy, but not for the test itself. So I think I always think of that, if you think of the boundaries of myeloma therapy and you put on the x-axis the duration, I think MRD will help us because it will limit duration of therapy once we get to a sustained MRD negativity with the right point. But also on the y-axis, it tells us about the depth of the response. And there's many features that we can translate that into. For instance, if you look at depth, it's prognosis. And prognosis is a very important conversation. Sometimes physicians say, what am I going to do different? And my response is, well, the first thing you do is you pick up the phone and you talk to the patients. And you say, well, you've been able to achieve this, and this is what this means long-term. MRD negative is now being synonymous with being cured. But it's a very important step for someone who's able to achieve that, to know that they have greater assurances that over a period of time, the myeloma will not come back. But not only that, if you remain positive, I think we should consider additional treatment. And the last target would be that if you remain negative and you're on chronic therapy, hopefully we can spare people being for years on maintenance treatment, such as would be with Revlimid, which we might think of it benign, but I think many of the audience know it causes fatigue, sometimes diarrhea, there's the cost, sometimes the fogginess, and all the things that come with chronic therapy.
To add to that too, like I've had friends who said, I just became MRD positive, and I think, I have kind of higher risk disease. I should probably start something. So, but not to wait till it keeps growing until I have this M protein to take early action. And I think what you're saying is really, really important, what both of you are really saying. It makes a lot of sense. Dr. Landren, now when you spoke with us a year ago in a roundtable on imaging with Dr. Hillengass, you raised a great deal of interest with your ImmunoPET project, which you planned when you moved to Miami. And I also learned, like we're talking about, that you're doing a study on why patients in remission might not need maintenance therapy. So can you kind of follow up with what Dr. Fonseca said on that topic, and then update on that as well?
Yeah, so we did develop, when I worked in New York at Sloan Kettering, the ImmunoPET, where we labeled actually homemade antibodies. We made antibodies for CD38, we had BCMA, and we labeled them and we gave them in the laboratory to mice that carried myeloma. We were able to show that these antibodies were detectable with the CAT scanners from mice. We could do all the experiments, and that really motivated us to develop a study where we could use antibodies also to go into humans. For practical purposes, we chose to pick one of the FDA-approved antibodies because otherwise you have to file the same work as you would do for a new drug. It's a lot of filing and a lot of data you need to generate. So we used daratumumab and we labeled that the same exact way. We did it first in mice, and then we were able to prove that it worked, and then we did it in humans. So the work we did was a phase 1 study where we showed how to do it, what dose, what interval between the injection or infusion of these labeled antibody and the imaging. All those things were sorted out. So as the extension now, we have a phase 2 study that we are opening. We developed this here in Miami. We had hoped to open it earlier this year in the spring, but we had to set up additional processes for the antibody making. I think the study will open, I'm not 100% sure, but probably in mid-October this year. And we will image people that are looking for therapy. It could be newly diagnosed patients. It could also be patients that are in the relapse setting. The study will allow us to do imaging with this antibody. It's about 1% of a treatment dose with a very low amount of antibody. And patients can then be also imaged with regular PET-CT. They can undergo testing with blood tests, the bone marrow testing, all the standard workup. Patient can be treated with either standard of care or on a clinical trial therapy. And patient can then be re-imaged. So it's a separate protocol that just refers to imaging with these labeled antibodies. So it's before therapy and after therapy. And the study is designed to see if this tool can identify disease that we would not capture with MRD assays in the bone marrow. We also have access to blood-based MRD tools and PET-CT and all the other standard tools. So it's really a way to leverage detection of disease. And the rationale behind this is simply because we know that when we do biopsies and patients are MRD negative, there could be individuals, but there could be pockets, small pockets of disease that we could see if we do advanced imaging with other types of modalities. We want to see if we could capture any such. The last part of this study is that if patients agree, we could also do something called image-guided biopsies. If we see that there are residual sites, we can take out those cells. And in my research lab, we are set up to do both single cell and whole genome sequencing. And we have published extensively on that in the bone marrow. So the idea here is to see if we treat patients and we can see there are some residual sites, do they have anything in common? And what do they actually show? Is there something that we can go after by adding some drugs to what we already do? So we're basically looking for the Achilles heel in these cells and the microenvironment. So that's how that work goes. And you asked me about the maintenance. We are studying patients who are reaching MRD negative, being on maintenance, and we have seen that patients could be MRD negative for several years. We see that if we have 2 years of MRD negative, but that seems to be a very strong predictor of long-term freedom from progression. So we don't know if patients safely can go off, but we have opened and we are opening more studies to look into this. So we don't know the answer to it, but this is an area we are investigating.
Yeah, I think it's such an interesting idea. When I was diagnosed, maintenance therapy wasn't really a thing. And so I went for multiple years without anything. And if a patient can do that safely, I think that'd be really, really important. Um, you talked a lot about, like the genetics too of the disease. And Dr. Fonseca, I know that the Scottsdale Mayo Clinic group is highly proficient at myeloma genetics. You're talking, Dr. Langren, about seeing the whole picture of the patient, the genetics, the imaging, just everything. I think now that all these immunotherapies are coming closer to frontline, we also have to ask about this immune status. So, Dr. Fonseca, do you have, or do you both want to weigh in? But maybe Dr. Fonseca first. How do we get better at determining which patients might respond to these newer immunotherapies?
That is a $64,000 question right now when it relates to the immunotherapies, right? Because we're just totally in diapers there. We know that by, just looking at the mechanism of how those immune therapies work, in many occasions, it does require the recruitment and participation of the other cells in our immune system that somehow had overlooked myeloma cells as being bad. Just for background for our audience, our immune system protects us against external enemies, if you may, bacteria, viruses, but also internal ones. Our immune system should be checking for cells that seem to be abnormal. And one of the normal functions of the immune system is to protect us against cancer. There are, in fact, some cancers that we're seeing an increased, prevalence of more likely to happen. For instance, in the age of the AIDS epidemic, when people were, developing cancers that we would normally not see, but we were seeing them just because the immune system of those people were, going down, and then the cancer could thrive. But taking it one step further, we ask the question now, okay, what are the requisites so that if we give someone an immunotherapy, that immunotherapy will work? And so using some examples might be illustrative to this. So if we talk about, for instance, let's say CAR T cells, right? So if you're going to take T cells from someone to develop a CAR T cell like product, the first question is, are those T cells good enough? I mean, do they have the strength to reproduce and to attack? Or, perhaps in some situations, could it be better to collect T cells earlier on and save them for a later treatment? Or maybe even better yet, maybe CAR T cells should be used right from the get-go at some point early in the course of the disease. The same would be true for other antibodies like, the bispecifics. The bispecifics really are just a soluble form, if you may, of CAR T cells. They're kind of matchmakers, or I call them double-sided Velcros. They just bring together T cells and target cells in our disease of interest, that is myeloma. But do we have enough of those cells, and do they react properly? Or if you give something like, even simple daratumumab, which I call simple, of course, there's been a revolution in our field, but simple because it is just what we call the simple antibody. Do we have enough immunity to make sure that that drug converts into response? So people are starting to study this, and there's a number of studies where they're doing profiling and characterization of the bone marrows. One could even ask, should we do that, for instance, of the blood, or even maybe the lymph nodes, so that we understand if the body is giving the signal that there's something wrong there, here's an antibody, there's myeloma cells, there shouldn't be there. How can we tell who's going to not only respond, but is going to keep that response? So there's several teams, our group, the one of Dr. Landren, I know there's some work going on at Emory and other places where they're looking at, let's understand the immune response profile. We're very fortunate that we have much better tools than we ever had in the past, where people can ask very broad questions and they get answers from everywhere, meaning all the cells that are in the bone marrow to tell you, yeah, it seems like this person might be better responding or not, but it's still very, very early on.
Okay, Dr. Langren, do you have any comments about that? At least it's recognized. That's a starting point.
Well, I do think for a long time people have recognized that there is more to it than just the myeloma cells. Rafael went over many very important aspects. To share a little bit what we are doing in this kind of context, we have done single cell sequencing of peripheral blood. We have also done these types of assays of the bone marrow. And we actually see the very similar patterns of skewing of different cells in individual patients in these two types of samples, which I think is very important because now you can do a blood test instead of a biopsy and you can study it over time. So that's obviously a huge practical advantage. We have also studied this, and we are about to present a paper at this upcoming ASH meeting, where we have studied these types of changes over time in people that were treated on maintenance therapy with lenalidomide. And we have studied 100 patients longitudinally with blood tests every 3 months, and the longest follow-up goes all the way up to 10 years. So what we have seen is, what we have focused on is patients that achieve MRD negativity and sustain their MRD negativity. And then we have compared that to people who did not reach MRD negativity or individuals who became MRD negative and lost it in a shorter period of time. What we see in the microenvironment, without giving away all the details before ASH here, so I steal my team members' thunder, but I can say that we see that there are actually big differences in the host immunity. And I think I can probably give away and say that there are signs of the immune system actually normalizing, without giving any details. There are signs of the immune system normalizing in those individuals that are able to achieve and sustain MRD negativity. So in my mind, I think the perfect combination of variables for a good clinical outcome is to have good therapy, to not have too aggressive disease, and to have a healthy immune system. And I think that's what this study is kind of showing. There are a lot of other groups. Raphael mentioned the Emory group, Dana-Farber. I think Mayo is working on this as well, and others, NYU also. Many other groups are working on this.
Mm-hmm. Well, what an important thing.
Can I throw in just a very quick comment? And this happens often with patients, that people will go and say, take this or that because it's good for immunity. So I'm going to put immunity this way. Let's say it's like, take this pill because it's good for your personality. I mean, what does that mean? What aspect of your personality, etc.? So I think people just need to be worried about that. I find that this is an area where people get a lot of advice that, even when it's well-intentioned, may not really be something that is going to be assisting or helping as they go through their treatments.
Yeah, there was a patient actually at my facility that was drinking one of those juices, those immune system boosting juices. And the nurse was saying the chemotherapy that they were using on the patient didn't touch it. Because they were using this product, and they finally said, "What are you doing?" And they confessed to drinking this every day. They just said, "You have to stop because our treatments are not working for you at all because this is happening." So it's true. I think we don't know enough. Well, we're getting close to our Q&A time, so I want to ask you a very important question. And Dr. Fonseca, maybe you want to start. I think a lot of patients want to know, with all these new treatments, especially some of these drug classes like bispecific antibodies are just exploding in myeloma. What excites you? How do you prioritize? What do you see as, and Dr. Fonseca, you kind of mentioned this at the beginning, that you think some patients are being functionally cured. So where are we at for this cure, and what is the most exciting things that you are seeing?
For me, you already mentioned, is the bispecific antibody, because the fact that we can have a medication that is going to be off the shelf, meaning I can write for it and an hour later a pharmacist could be mixing it so that it's given to the person without any wait, and potentially with similar effect as what we could see with CAR T cells, is incredibly exciting. And I drive this excitement because last year there was a paper that looked at the treatment of a form of leukemia that's called ALL, where they use a bispecific antibody, and they use molecules. A small molecule comes in a pill form for the treatment of that leukemia. The drug is called dasatinib, which, by the way, doesn't work for myeloma, but works for leukemia. And they were able to show the best outcomes as what you get with chemotherapy and with transplant. So the fact that they have been able to move beyond chemotherapy, and to do so with a very rational combination with an antibody like a bispecific and a small molecule, was fascinating. So my hope is that we get there at some point very soon for myeloma, that with combining, bispecifics and, some of the things we currently use to be able to make that a reality for many patients. My hope is also one of subtraction. So I would like to subtract the stem cell transplant from the future of myeloma patients being treated. I would like to subtract either the side effects or the drug altogether, the dexamethasone from,
Yay.
I like to subtract peripheral neuropathy from the life of myeloma patients, so that a colleague of mine who works in another area told me, you guys in myeloma are either a drug away or a combination away from curing a large fraction of myeloma patients. So that's my hope. One of the challenges is that once you have a treatment that is very effective, it's harder to take away. And we see this with children leukemia. Children leukemia, it's like, I don't know, 80, 90% curable. So who wants to mess with that? You don't want to change that. So I hope we can get smart enough to get there without having too much in our menu.
Mm-hmm. Yeah. Okay. Great answer. Thank you. And Dr. Landren?
I share the same sentiment as Raphael. I think the bispecific having off the shelf looks fantastic. Looking at all these different drugs where we currently are is early on. They're small series, they're short follow-up, but doing extrapolations of these drugs compared to all the other already FDA-approved drugs, to me, they look like a magnitude of 4 to 5 times more effective. They are subcutaneous, many of them. I think and I hope that the dosing schedules will be with long intervals over time, and I think also paired with sophisticated blood-based MRD testing. I hope and think that there could be patients taken off these therapies and monitored safely, as we discussed before. I know that all these pieces have to be validated in order to become like truths, but I think these are hypotheses that I think we will test in studies. We're going to move this fast forward. There are so many drugs in development so that there will be winners. There is no doubt about it. Mm-hmm. I think also when it comes to CAR T cells, I think different patients will have different perspectives. So I don't think that CAR T cells will not be relevant. I do think they will have a role also. I think for patients who are suffering from later relapses, where the disease comes back after very many lines of therapy, the fact that the patient is receiving a lot of therapy frequently, all patients know what I'm talking about. All doctors who treat myeloma know what I'm talking about. So having a break from all this is huge. So I think if there is a therapy that can give a longer interval without having to come back and get infusions and drugs all the time, I think there will be many patients interested in that too. And then, of course, long term, if it turns out that one or the other has, like, if there is a curative component to it, I hope that some of these modalities maybe that will eventually be the big winner. But I think until we are there, I see that the cell therapies will happen and they will stay. I think the antibodies will happen in terms of development. They will live in parallel. I think the high volume, I think it's going to be antibodies because of availability and combination opportunities. But I think that will be a combination of both. I don't know what Raphael thinks about that.
I agree completely. So I think combination and ideal combination will be the key for success.